it's my particular pleasure to host this next set of short talks. So over the next 15 minutes, we will hear short presentations from 3 participants in the 2021 JGI-UC Merced Summer Internship Program. The JGI has partnered with the University of California's newest campus in Merced in the Central Valley of California for the past 7 years. And so each year we bring both undergraduate and graduate students to the JGI for the summer. And here at the JGI, they work side by side with JGI mentors on actual ongoing research projects. And we have quite a few. Today we will hear about some projects from just 3 of the students who were at the JGI this summer. The first presentation will be given by Maria Mendoza and Lorenzo Scaturchio. And they've been working on different aspects of the same project, so they will give a tag team presentation. Maria is a 4th-year grad student in the Quantitative and Systems Biology program, and Lorenzo is a 4th-year undergrad in Science and Engineering. Before I turn it over to Maria and Lorenzo, I just want to point out that in the interest of time, we will not have a Q&A for these very short talks, but you can use the contact feature in the meeting platform to get in touch with them after the presentations. And with that, I'll turn it over to Maria.
Well, it's a pleasure to be here today. Flowering plants are divided into 2 types. We have dicots. This dicot root belongs to the model organism Arabidopsis. Deep knowledge about plant genetics comes from this plant. However, bioenergy crops like sorghum, miscanthus, are monocots. And we can't continue to use a dicot as a model organism for monocots. Additionally, in order to better understand how these plants grow, it's to study them at the single cell level. We decided to use rice single cell data to improve species comparisons of single cell data sets. Here we compare the dicot Arabidopsis to the monocot. So how do we obtain single cells? Plant cells have cell walls, and we have to get rid of them in order to perform single cell RNA sequencing. We can do this through an enzymatic digestion, and after incubation, you're left with protoplasm. And these are just plant cells with no cell wall. Once you have your individual cells, you can follow. The primary goal for this technology is to code every RNA transcript in every cell. Each barcode is unique. You can start by flowing your cells through one channel and your barcoded beads through another. On the right, you have oil. So when the cells and the beads meet, an emulsion droplet encapsulates them. The library is created and sequenced, and when you've processed your reads, you end up with this count matrix, which you can then use for statistical analysis. Then you can cells into clusters based on the expression patterns. Once you've sequenced the transcriptomes of your individual cells, you can use a reference data set to learn more about your query data set through a process called integration. And this is when you combine 2 or more data sets into a shared space for joint analysis. One of my mentors this summer, Benjamin Cole and his colleagues, created a reference data set for the Arabidopsis root last year. It has single cell resolution, so we integrated the Liu et al. Rice data set to that reference data set, and this allowed us to transfer information from the reference to the query. Our goal is to perform 2 types of integration. An intra-species integration between 2 different types of rice, and an interspecies integration between rice and Arabidopsis. Here we have the individual UMAPs for each data set. A UMAP is the definition of it, but it's used to visualize highly dimensional data sets. Here we have our integrated data sets colored by cluster. And we can see that both the intra-species and interspecies integrations have well-defined clusters of cell types, and that's great. Here we have our integration or our integrated data sets colored by species. And we can see that the intra-species integration has a lot of overlap. And this means that it's really well mixed, and that's fantastic. However, our interspecies integration didn't have almost any overlap, and that means that it is not well mixed. In summary, we learned that intra-species integrations are easier and interspecies integrations are more challenging. We'd like to continue to optimize our interspecies integration using alternate methods. We would also like to score these integrations so that we have a quantitative to better compare the different methods. But my partner Lorenzo will talk to you more about that.
Thank you, Maria. So what we wanted to develop was a way to evaluate the quality of integration quantitatively. So we selected this specific pipeline. So the first step would be to take the input data and run it through RNA sequencing workflow. Then there's batch effect correction, which are variable factors that contribute to an error in analysis, and they're important for cross-species analysis. So one form of correction for these batch effects is integration itself. So this results in our final scoring methods of both k-nearest neighbor purity and entropy of batch mixing. However, we first want to understand what integration looks like. So looking at the first figure on the right, we can see that each shape and plane represents the type of species, while the color represents the cell type for that particular species. When looking at the integration process, we can see that the clusters are intertwined. So this results on that right side figure, all the clusterings of each type of species. And what this basically means is we're able to compare both Arabidopsis and a rhizos homology in a similar fashion. So entropy of batch mixing. Entropy of batch mixing asks how many data sets are in the vicinity of a given cell. So this is a measure of data set and mixing. As we can see for the low score, they're isolated from each other and we would consider not mixed in. But in the high score, we could see better mixed batches. K-nearest neighbor purity, on the other hand, is scored on different types of cells in the vicinity of each cell. This is also known as the measure of cluster purity. So in the low scoring figure, the cells are less separated and are latched onto the clusters, while on the high scoring figure, you can see the intertwined cell types have more isolation and distinction. So this will yield a higher score. So here is the intraspecies integration of both the rice cultivars of Mendoza and 9311 rice variety. On the left figure, you can see that color, so cell type. And on the right side, it is integrated by species. So it's colored by species. And on the left side, you can see that the clusters are very distinct and isolated from each other such that it would indicate a high cell type entropy score. While on the right side, you can see there is significant overlap with each of the batches, which would indicate a high batch entropy score. This all has to do with the idea that we're comparing two rice cultivars together. This is the interspecies integration of both rice versus Arabidopsis. So as we can see, things are very similar. It's colored by cluster on the left-hand side, and on the right-hand side, it is still colored by species or batch. On the left-hand side, once again, you can see here that the clusters are somewhat distinguishable. However, there's mixture between the cell types, and therefore it can be a little bit fuzzy to indicate. Therefore, we would say this is a moderate score for cell type entropy. On the right-hand side, you can see there's little to no overlap, and therefore we can determine that this batch entropy score would be low, considering that we can't see any of the cells overlapping except for certain areas. And this just has to do with the idea that we are comparing two different types of species. A lot harder to do. So to conclude, we analyzed clusters of Arabidopsis and two cultivars of rice, created a one-to-one homology list for cross-species comparison, integrated single-cell data sets of two species of plants and two cultivars of rice, and there's a strong need and desire to create M-to-M homology list using the single cell data. Both Maria and I would like to thank Margo Bezrodzik and Axel Wiesel, for everything that they've provided for us and the Interns. Thank you.
Thanks, Maria and Lorenzo. That was awesome. I saw some comments in the chat. We had some sound issues, but I hope you could still understand everything. It may be because there were two presenters at the same time. So hopefully this will not be a problem for the next one. So in the interest of time, we'll go straight to the next presentation. And this will be given by Beryl Arinda. Beryl is a second-year PhD student in the Quantitative Systems Biology program at UC Merced. And over the summer, she worked with Matt Mingay and Sharon Greenbloom here at the JGI. And she will now tell us more about what exactly she did during that time. So Beryl, take it away.
All right, thank you, Axel. Allow me to shift our minds from rice and Arabidopsis and introduce Panicum Hallii. Panicum Hallii is a model crop commonly used in studies about perennial grasses, and specifically to study its genetic relatives, pear grass or Panicum virgatum. This is of economic and industrial importance because it's used as a biofuel. So any genetic studies could lend insight on genes needed to introduce helpful properties like drought resistance, antimicrobial resistance. So in the JGI lab at the University of Texas, Panicum Hallii plants were exposed to fast neutron irradiation. This technique introduces a wide range of random mutations, and this could reveal unknown genes or impact of mutations on known genes. So the output of the sequencing experiments is stored in a variant call format or a VCF file. In the initial data set, we had 89 samples with over 3,460 variants. But the entire pilot study had 1,000 samples. So we expect more variants, not just from the pilot study, but exponentially more when working with bigger sample sizes. So the VCF file is extensively detailed, things like chromosomes, variants, type of mutations that are introduced. And so this is great because it has all the information that a researcher may need, but it's challenging because you either need to have programming knowledge and skills to maneuver this information or the time investment to learn. So this is where a partnership with the user support analysis group at JGI comes in. Our goal was to design a tool that can be used to maneuver the information contained in this data file. We use bioinformatic tools like SNPF to annotate and predict effects of the genetic variants on genes and proteins. For example, if there are any amino acid changes. We employed use of Python, R toolkits like dplyr, and also to build a variant explorer tool, we used Shiny, which is an R package that makes it easy to build interactive web apps. Let us look at a brief demo of the variant explorer. If a researcher is interested in a gene associated with a known function, they can input the description of the gene based on this function. For example, ubiquitin, which is a regulatory protein. The output will show a list of genes associated with a function. So also if a researcher knows a specific gene they're interested in, either from the search or literature review, they can input the gene ID, and the output will include things like the list of samples, mutation types, if any, the location of the gene, chromosome position, and can also be filtered and a count performed of how many samples had this specific kind of mutation. Also, if our researcher is interested in a specific phenotype, maybe something that they saw in the plants, using keywords, common word descriptors like whitish, pale, this is usually associated with chlorophyll irregularities. The images of the phenotypes show up, and you're able to also see details of putative impact of the mutation. For example, if it was a large chromosome deletion with high impact, meaning it disrupts the protein. So here we have a simple user-friendly tool that is dynamic and functional, which is what we set out to do. It can be used to maneuver through a large data set and can be used to discover gene candidates for future studies. So we believe that we now have a study framework for a usable tool, which you can build on to add more functionality and increase the tool's scope. I'd like to once again thank my mentors, Matt and Sharon, as well as the team at JGI that supported our internship to run smoothly. Finally, the academic support of my advisors and department at UC Merced. Thank you.
Thanks, Beryl. That was great. Unfortunately, we don't have an applause feature in the platform, but I'm sure the rest of the audience was just as impressed as I was by the presentations by the three speakers and to see what they accomplished just over the pretty short course of a summer.